Hi everyone, good evening. So this class I'm taking it is based around industrial and different productive uses of microbiology. So of course one of the main uses of microbes in producing a product that's useful for human society, or it's being used in human society, would be the production of alcohol, because we have this fermentation as one of the major parts of beer production or any of these other beverages. Fermentation plays a major role in a lot of these foods and drinks. So this part of the notes is literally on beer. All right. So first slide that the professor marked. It's part of the economy. It was found in Sumeria. We have this artifact that took place 2050 BC, and it was a receipt for beer. So this beverage was a large part of the economy, even in the earliest civilizations. Right now, we have a volume of beer worldwide of 1.89 hectoliters, 1.89 billion, I should add. There's a difference there. 1.89 billion hectoliters. The word alcohol actually comes from al-ghul. It's Arabic in origin. Al-ghul means the cover-up, and then alcohol. This is kohl, which is the eye paint produced by sublimation. This is similar to distillation. Okay, so water is the main ingredient in beer. Sources include surface water, which is from lakes and rivers. Groundwater is from wells. And then municipal water is a third option. Characteristics wise, water used, it's free of odor. You have anions. You're going to contribute anions. You're going to use chloride anions. This will give a fuller and a sweet taste. And you're also going to have sulfate. This is going to make a drier and bitter beer. Cations are also going to be introduced by water, and calcium is going to add clarity and stability, while magnesium is going to help with pH of the mash. Hardness is going to be important in controlling pH. How you're going to adjust the chemistry, you're going to be adding calcium chloride or sulfate or sulfuric acid to again control the pH of the beer. Another main component will be the plant Humulus lupulus, also known as hops. These are the flowers of this Humulus lupulus plant, I should say. Only hops from the female plants are used, and each cone has these lupulin glands. They add bitterness and aroma to the beer. The composition includes alpha acids. During boiling, these alpha acids will isomerize to iso-alpha acids. There are also beta acids. These are going to be contributing to the aroma. And then you have essential oils. These are going to be the terpene hydrocarbons. They're going to add piney, floral, or spicy and earthy, aromas, for lack of a better word. Okay, let's look at the yeast biochemistry. The sugar sources for yeast will be malted barley, corn, or rice. From these sugar sources, you're going to get our pyruvate, and then you're going to get acetaldehyde, and then this is going to go to ethanol, which is, of course, an alcohol, and then your CO2 during the actual fermentation. You're going to first go through the brewing process. The first part will be, of course, malting the barley. So barley is going to be steeped in water. You're going to allow them to germinate with air, and then the starch degrading enzymes will start showing up. We have alpha amylase and beta amylase. Then you're going to kiln the barley for 4 to 5 days. Okay, let's look at the malt color and the flavor. So The drying temperature as well as the time it's being dried at, this will affect the color and the flavor of the malt. The darker they are, the stronger flavor they have. A pale malt will be low temperature. This is like Miller or Jaeger beer. And then brown, this will be a pale ale or brown ales. And then dark malts will be for stouts. What happens would be you're going to have the pale malt, and then you have little amounts of either brown or dark malt. Next step will be the mashing step. This is where you're going to grind up the malt. You're going to add water at 148 to 152 degrees Fahrenheit, not degrees Celsius. That's a little too hot there. 152 degrees Fahrenheit. There we go. This is going to convert starch into sugar. You're going to also have adjuncts like rice and corn. Adjuncts will be used to add starch that could be converted into fermentable sugar. Let's look at a mash scheme or a mash schedule. So a mash scheme will show this graph. Basically, you have temperature and minutes on the x-axis. And the data that we're going to plot would be these two lines. Let me make one of them a different color. Okay, so that purple line I just drew and that green line that was already there. So lower fermentable sugars are going to be produced by favoring the alpha amylase. At the higher temperatures of 157 degrees, the lower fermentable sugars, they are going to be a little bit sweeter. This is how you get a sweeter beer. The higher fermentable sugars are going to be produced by beta amylase. These are going to be found at temperatures around 144, so right here. And this is going to be more alcoholic of a beverage if you're playing around with the higher fermentable sugars. So the time and the temperature are going to affect the fermentability of the wort, as well as the beer sweetness and alcohol content. This is the next step, the straining or the lautering step. The mash is heated to 170 degrees Fahrenheit, not Celsius. There we go. And this is going to denature enzymes. You're going to filter through stainless steel mesh. The grain is sparged with water to remove the sugars. And you're going to use this husk as a filter medium. When we call the clear liquid, when we want to call it something, we're going to call it the wort. This is the wort and the clear liquid that comes off of this lautering process. Alright, now we have a step of boiling. So this is going to prepare the fermentation. We're going to be adding the hops at the boiling stage. We're going to boil the wort, and this is going to extract a bitter, like floral aroma from the hops. This is also going to sterilize the wort, and it's going to also precipitate these proteins and polyphenols. Okay, post-boiling wort clarification. The denatured proteins and polyphenols are also known as trub. Let me see where I wrote that. There we go, trub. And you're going to remove the trubs by settling in a hot wort receiver. The tangential flow causes a pile of this trub at the center, so you're able to just clear it up very nicely. Let's talk about fermentation and the idea of pitching the yeast. This is when you're going to add it. So in ale, you're going to be using Saccharomyces cerevisiae. This is going to be a top fermenting species at 70 degrees Fahrenheit. Weiss beer gives it a clove-like flavor. So this Weiss yeast will give a clove flavor. In lager beer, you're going to be using Saccharomyces pastorianus. And this is a bottom fermenting yeast at around 50 to 55 degrees Fahrenheit. Oxygen is only added to the wort at the beginning while it's being filled. This is used for the yeast to grow. And you're going to also add some sterols for the plasma membrane. Fermentation, however, of course, is anaerobic, and the carbon dioxide made will agitate the system, while the cooling jackets will help with temperature control. So again, the two yeasts, so ale, it's Saccharomyces cerevisiae, lager beer, it's going to be Pastorianus. Alright, maturation, the next step. This is going to reduce the acetaldehyde or a green apple taste, and the diacetyl, which is going to be like a butter taste. Ales, the temperature will rise from 72 to 75, and this is going to be 1 day for maturation. However, lager beer will be cooled to 45 degrees Fahrenheit, and this is going to take 3 weeks in order to help with clarification of the lager beer. We also have some post-fermentation treatments. We could add some dry hops. So dry hops will, again, allow for the aromatic level to increase, as well as the citrus and pine notes. IPAs are going to involve the dry hopping. Alright, now let's talk about, let's see, where's the next part? Okay, so now it's just the ingredients and their flavor contribution. The next part I accidentally wrote in the notes even though we don't need it, so I'm just gonna cross that part out like so. Okay, so the ingredients and their flavor contribution. So hops, we were talking about, these are like the aromas. We got a floral aroma as well as a bitterness taste. Malt is going to contribute the caramel color as well as a malty feel or malty taste. Yeast will allow for the fruity, grassy, butterscotch, sulfury, and alcoholic taste. Again, we don't need the types of beer chart. That was crossed off. I don't know what this 4.6% ABV was. I think that's beer's like typical range, but we don't need that either. You change the notes. Okay, so quality assurance in the beer industry involves alcohol component, apparent extract, real extract. Apparent is like the gravity of the finished beer, indication of sweetness. So apparent is the indication of sweetness. However, the real extract is the finished beer adjusted for alcohol. Bitterness is also checked, CO2 content, the fizziness, the foam, diacetyl level, the color level, free amino nitrogen. So again, the amino acids from the growth. For the yeast, the pH is also checked, and you also look at the haziness. Some beers do want a little bit of haziness. Right, that's all from there. Please do something nice for someone, drive home safely, and yeah, have a good one.